Good evening, Mr. CTO. This proposal is about using data science for drug design and clinical outcome prediction for personalized cancer immunotherapy. This proposal has been approved by Pfizer CEO, but we all know without your approval and support, this proposal will not go anywhere. So we ask for a hire of 8 data scientists who have immunology background for an annual budget of 0.8 million at first phase. Half the data scientists will focus on rational drug design. The other half will work on predicting clinical trial response from patients. This proposal is based on the following facts. First, Pfizer is eager to become an impactful player in cancer immunotherapy. This is a new rapid expanding oncology branch for cancer treatment. The current cancer immunology therapy drug design lacks a rational approach. It is still based on experience and traditional trial and error testing. We all know deep learning is a powerful tool to capture non-linearity. In immunotherapy drug design, molecular interactions are highly context-dependent and non-linear. So our hypothesis is deep learning can play a big role here for rational drug design. Deep learning has huge potential in predicting clinical response from patients taking certain drugs, and ultimately will enable us to prescribe personalized therapeutic medicines for each patient. Before going to the details about this proposal, I'd like to share with you a little bit about cancer and immune system. So we all know cancer is caused in part by mutations in DNA sequence. This mutated DNA will generate mutated proteins in the cells. An adaptive immune system will present those mutated and foreign proteins in cancer cells on cell surface. Normally, T cell will recognize those foreign antigens and kills those cells. And this will lead to T cell expansion. However, in cancer cells, cancer generates some specific signals, and those can shut down the activity of T cells. Checkpoint inhibitors are the most successful type of immunotherapy for cancer so far. Basically, the main function of that inhibitors is to relieve the inhibitory effect on T cells, so to reactivate T cells, so T cells can do their job and kill those cancer cells. With too many T cells activated, they can sometimes cause problems. So the main side effect is the autoimmune activity from the T cells. And also, not every patient is going to react positively with immunotherapy because some cancers have more mutations than the others. And not all patients have active and available T cells to fight with cancers. And there's also other unknown factors. So the proposal 1 for this proposal is to predict immunogenicity of antigens. To do that, we first have to predict the interaction between the fragment mutated protein fragments and MHC complex on the cell surface. So we have to model interaction between MHC protein and the fragment from mutated cancer cells proteins. The second prediction is going to be to predict the interaction between MHC and mutated peptide, or sometimes called antigen interaction, with the receptor on T cell surface. So the ultimate goal is to determine which immunotherapy drugs likely to be most effective based on the first two predictions. For MHC binding prediction, so here is the protein structure for MHC, which is about 200 to 300 peptides. And this is the very short peptide, but normally it's about nine amino acids. These peptides from the mutated proteins inside cells. So there are thousands of MHC anneals in human population. Each anneal is capable of binding to a distinct set of peptides. So our objective is to predict whether our MHC anneal will bind to a given peptide. So far, there's a lot of public data available. Those are based on experimental results. So those data going to show you which anneal going to be interacting with which peptide and how strong their binding going to be. For example, here 5,000 obviously is a strong binding between these two parts. 20, that means it's a weak binding between these two. Also, there's other public available data which regarding to the interaction between T cells and MHC antigen complex. So as you can see here, sometimes the interaction is negative, so this T cell doesn't recognize this MHC antigen complex. Sometimes it's positive. So there's a lot of public data available. So we can use those public available data to train our model. So I use an example from the website to show you we can do machine learning and to predict what type of antigens are presented on the cell surface by MHC. The MHC anneal we choose is HLA-A0201, and MHC peptide binding data was got from a website. For training, we do cross validation. We divide the data into training data sets and then test data, which the ratio is 9:1. So the results will be shown based on the test data sets. Originally, some researchers carry out the prediction. They used CARES packaged in our environment. We were able to reproduce the results with TensorFlow in Python environment, and we confirmed the high prediction accuracy at 95%. From training data sets using our model, we saw that only selective peptides will be presented by MHC anneal. That means not every fragment from those mutated protein in the cancer cells will be presented on the cell surface and to be recognized by T cells. As you can see here, position 2, position 9, they prefer to very specific type of amino acids. And using feed-forward neural network, we got an accuracy of 94.8%. As you can see here, zero class means no binding between MHC and peptide. 1 means weak binding between these two, and 2 means strong binding between these two. The blue dots represent the correctly predicted events, and red dots represent incorrectly present events. We also used convolutional neural network, and accuracy is pretty similar, 92%. However, when we tried the random forest model, the accuracy dropped a little bit from 95 to 82%. This suggests that use neural network probably is a better option for modeling the MHC and peptide interactions in our case. So a quick summary for proposal 1, predicting immunogenicity of antigen. Although I didn't show data that we can do prediction between this MHC and antigen complex with interaction with the receptor from T cells. But I hope I can convince you that use data science and correctly use the models, we can to achieve our ultimate goal, and that is to determine which immunotherapy drugs likely to be most active for each cancer patient. Mr. and Jen are going to be present the proposal 2 for you. Thank you very much.